Chapter 2 will take us through the first level of organization found in the human body, the chemical level. A lot of the information presented in this chapter will be very familiar to most of you. We'll be borrowing from a lot of concepts from both biology and chemistry from high school, and we will touch upon some new things such as biochemistry, although not in quite a lot of detail. That's pretty much a course all in of itself. As we move through the chapter, we'll apply it as best we can to human anatomy and physiology. In this chapter, we'll go over the fundamental concepts of chemistry. We'll talk about how matter is organized in the human body. We'll talk about the three prominent chemical bonds found through chemical reactions. And finally, we'll compare and contrast both organic and inorganic compounds and how they're used fundamentally in the biochemistry of the human body. So the basic principles of chemistry, of course, it'll look at the structure and the interactions of matter. As we know, mass is matter that takes up any kind of space. Where mass is the amount of matter found in a particular substance, the weight is going to be determined by the force of gravity acting on that mass. That'll be very important when we talk about bone and muscle in particular. So look at how gravity will interact, or lack thereof, will interact with certain types of tissues, and we'll see how the body will manifest itself in different types of environments. And of course, as we know, matter is found in three forms: solid, liquid, and gas. Some of you will throw in plasma here as well, but for the most part, we'll be dealing with these basic forms of matter. And of course, all forms of matter can be composed of different chemical elements. We'll be focusing mostly on the organic elements, also the lesser known or the trace elements as well. But for the most part, we'll be talking about carbon, hydrogen, oxygen, and nitrogen. So those four compounds or elements we found in the body will make up the majority of the mass in the human body, about 96%. By weight, oxygen is found to be the most prevalent, followed by carbon, hydrogen, and then by nitrogen. Less abundantly, we find the lesser elements, which take up about 3.6% of the body mass, which include things like calcium. It's very important for both bone and muscle tissue. Phosphorus found in bone, as well as ATP and also nucleic acids. Potassium, will find this in the intercellular fluid, very important for action potentials as well. Sulfur found in vitamins and proteins. Chlorine found in the extracellular fluid. Magnesium for enzyme function, and to some extent also bone properties as well. And of course, iron for hemoglobin. It is helpful to know which one is paired with which. You may see a question or two on these, but these will be reinforced as we go throughout the semester. And finally, to round things out, we have the trace elements of the human body, found at about 4.4% of the total body mass. Things like aluminum, selenium, silicon, copper, cobalt. A lot of these have very important functions with different types of enzymes, with immune system response. Some are very helpful for maintaining proper tissue balance for bone tissue, for nerve system tissue, as well as muscle. So atoms, as we remember back from high school chemistry, smallest units of matter that retain the properties and characteristics of an element. We'll talk about the protons, neutrons, and electrons, basically how that affects molecular weight and how it affects different charge properties found in ions in particular. And as we know, the protons and neutrons will form a nucleus, and electrons will form a shell as they orbit around the nucleus. So the atomic number, as we go through the different terms found in chemistry, the number of protons will be your atomic number found in the nucleus of the atom, whereas the mass number is the number of protons and neutrons found in an atom itself. Now this is based on an average of how many neutrons are actually found within the atom. As we know, there are different isotopes found with different numbers of neutrons, but again, we take the average to calculate the atomic mass. And we use the measure of Daltons to give us the idea of how much mass is found in a particular property of the atom. Neutrons and protons are relatively the same, 1.008 Daltons for neutron, 1.007 Daltons for the proton, and electrons coming at a 5/10,000th of a Dalton, minuscule compared to neutrons and protons. So naturally, neutrons and protons will take care of the majority of the mass found in the atom itself. So we calculate the atomic mass, or the weight of the element, is the average mass of all the naturally occurring isotopes. Now we will revisit the Daltons later on, especially as we get into the integumentary system and how that interacts with what kind of things can pass through the skin. In fact, you want to pay attention to that. That will be answered to a question later on down the line, both for your homework and possibly during the exam. And of course, ions will have a charge, either positive or negative, as they lose or gain an electron. A molecule will be two or more atoms sharing electrons, usually in a covalent bond. And a compound, where will be a substance that can be broken down into two or more different elements. So the very simple things like, say, table salt, sodium chloride, looking at nitrous oxide, nitric oxide, which is also found in the body. Look at a couple other massive compounds as well as we go into the macromolecules like glucose and different types of proteins, amino acids, lipids, and so on and so forth. As in Chapter 1, there will be a handful of animations found within this chapter as well. Those of you who have the Wiley Plus subscription, you can click through these on your own through my lecture PowerPoint slides, or you can go right into Wiley Plus in Chapter 2 and go through the animations, which I feel the animations are a nice little concise way of going over everything that we find within the chapter. It kind of brings everything together, and plus it gives you a nice little visual to go with some of the slides we're showing here. So chemical bonds simply occur when atoms are held together by forces of attraction. Now this has to follow the octet rule. So once we fill the first valence shell, which only holds 2 electrons, we go to the second valence shell, which will follow the octet rule and try and fill up all 8 positions for the electrons. Now carbon is very important for the human body because it has 4 electrons found in the outermost shell, which means it can bond with 4 other atoms at the same time. This is very important for forming different kinds of carbohydrate rings, and forming different chains of amino acids, so on and so forth. So the atom in particular will try and become as stable as possible and try and fill those electron shell as best possible to keep it most stable. First, we'll look at ionic bonds. Very simply, a positively charged ion is known as a cation. A negatively charged atom is known as an anion. So very simply, if you look at the uppermost portion here under sodium, if you notice it has 3 valence shells, has 1 electron in the outermost shell, so they can easily donate that to another atom, in this case a chlorine atom, which has 7 valence electrons, which will fill the octet rule by accepting an electron there as well. So what that creates for us is a positively charged sodium cation and a negatively charged chloride anion, and that will provide the bond you'll see here. So we see electron donation with ionic bonds. With covalent bonds, we're going to see sharing. You're usually going to see at least 2 electrons for each bond in particular between 2 molecules. So in this instance, for a single bond, you're going to see 1 pair of electrons paired between 2 hydrogen molecules to form hydrogen, so that will fill that particular valence shell of 2 electrons. If you look forward to letter B on the oxygen atoms, you very simply have 6 electrons in the outermost shell, looking for 2 to make it most stable. So very simply, 2 oxygen molecules will bind together, forming a double bond and sharing 4 electrons, or 2 pair, to form oxygen. And finally, you see on the nitrogen atoms only has 2 on the outermost valence shell. So between the 2 to make it most stable, we'll have a triple bond, which will have 3 pairs, total of 6 shared electrons. That will fill the octet rule and make this molecule very stable as it forms nitrogen. Now for 2 substances in particular in the human body for covalent bonds, one in particular is the carbon atom, which we can see in the outermost valence shell has 4 electrons in the shell, which means it can bond with 4 other potential molecules in this sense. So here we see 4 hydrogen atoms will bind with the carbon atom to form methane. So very simply, you have a single bond. You're going to see sharing of 4 electrons, 1 from each hydrogen, which forms up with the carbon to be very stable at that point. Now, this particular process is very important because this particular bonding with carbon will allow different types of chains to form, different types of sugars, whether it be pentose or possibly hexose rings, also very important for dealing with cholesterols and other types of steroids, and also forming different types of amino acids. It's this bond or this acceptability of the electrons from the carbon atom and also from the hydrogen atoms to form these long chains. Occasionally you'll see double bonds in some of the carbon atoms, which we'll look at, especially get into lipids, and whether we deal with both saturated or unsaturated compounds. Another type of covalent bond you'll see too is a bonding of the oxygen atom with 2 hydrogens, where what winds up happening is the oxygen atom will take the 2 electrons and not quite donate an ionic capability, but will actually hang out more so or closer to the nucleus of the oxygen atom. So this creates a polarity on the water molecule. So naturally we have a negative pole and a positive pole. So what we'll see is the negative pole will be closer to the oxygen nucleus and more positively oriented at the edges of both the hydrogens. This is very important, in particular when we talk about water as an inorganic molecule, we talk about different properties of H2O, it will be because of this polarity. So with the polarity, we see the third type of bond known as hydrogen bonds. So basically 2 other atoms associated with a hydrogen atom. So in this case, the negative end, just like a magnet, will attract a positive end. Now, a hydrogen bond by itself is relatively weak compared to the covalent and the ionic bonds. However, it's very important for dealing with three-dimensional structures, and we see quite a few of these hydrogen bonds found in enzymes as well as DNA as well as other structures. So when used in abundance, the hydrogen bonds are very stable and very strong. And of course, chemical reactions we see happen in both directions. We either see new bonds are formed or we see the bonds are broken. But very simply, the reactants will be your starting substances and the products may be your ending substances. And oftentimes, these will be interchangeable from both directions of the actual reactions. And the amalgamation of all the reactions found in the body is known as metabolism. You should remember this from chapter 1. So that's a combination of all the anabolic reactions and the catabolic reactions. And of course, this also follows the laws of the law of conservation of mass. So mass is neither created or destroyed. Different types of energy that we find will be potential energy, which means the energy stored in an object based on its position. Kinetic energy is the energy of motion, and of course chemical energy, which we'll be talking mostly in this particular chapter, is found within the bonds between different atoms and also the large molecules as well. This also follows the same kind of law of conservation of mass. Naturally, law of conservation of energy means that energy is neither created nor destroyed. When we talk about different types of reactions, we're going to look at either exergonic or endergonic reactions. Exergonic very simply means that you're releasing more energy that you're taking in. Endergonic means you're not losing as much energy as you're taking in. And a lot of times an exergonic reaction may fuel an endergonic reaction and vice versa. The other term to look at is called activation energy. This is the investment of energy that you'll use to drive that particular reaction. And we'll look at a special process in the next slide to make this activation energy make the whole process a little more efficient. So here we use catalysts. So as you can see from the graph here, very simply, the activation energy not using catalysts is very high. However, to make things more efficient, using a catalyst will actually drive the potential energy needed down. So very simply, we'll progress with the reaction, and therefore, we can use more energy over a multitude of different types of reactions all throughout the body. Different types of chemical reactions we'll find in the body include synthesis, which is anabolic reactions. These are basically building things from the smaller atoms and the molecules into bigger molecules. So think of amino acids building up into proteins and therefore enzymes. The opposing that would be the decomposition atoms or catabolic style reactions. These are simple things like taking in sugar and like table sugar, which is sucrose, and then breaking it down into fructose and glucose. You can have exchange reactions, where you see oftentimes, especially with the bicarbonate buffer system, found to try and balance out pH in the blood in particular. So you see hydrogen ions will bind with bicarbonate ions to form carbonic acid and vice versa. You often will see other reactions that are reversible. So these kind of things may include either exchange reactions or synthesis or decomposition reactions where very simply things will go in a to and fro type of thing or seesaw effect. Occasionally, though, you will have irreversible reactions, which we'll talk about briefly in the next lab. A lot of times, if you have an excess of heat and or too much out of the range of the pH balance, a lot of times certain types of reactions can run amok one way versus the other, and therefore the actual products can be irreversibly changed and not be able to revert back to the original form. And finally, we have the oxidation reduction type reactions. Of course, oxidation is when you lose an electron. Reduction is when you gain an electron. We see this with the electron transport chain in particular. And again, we also see this with other types of processes, in particular up and down the cell membranes. Inorganic versus organic compounds. Very simply, inorganic compounds usually lack carbon, are very simple. These are your table salts. These are your biosalts. This is water. This is oxygen, that kind of thing. Water is probably the most abundant and most important compound because it works as a universal lubricant and also a universal solvent up to a point. At least as far as the body goes, it is used for dissolving quite a different amount of substances. It's also where most of the reactions take place in some kind of water medium. The organic compounds, of course, will have carbon. Usually it will contain hydrogen atoms as well as oxygen, as well as a handful of others. And they always will have a covalent bond. So here's a nice little animation dealing with the polarity and solubility of molecules, which we'll test as well when we get into the lab. Once you're done looking at the animation, we'll see you at the next slide. So again, the polarity of the water molecule is very important, how well it functions and due to certain properties of water. Very simply, just having the negative and the positive poles here makes the water molecule very good for cohesion, to form hydrogen bonds. Was also very important for helping dissolve certain types of salts in particular, as well as some other substances, which again we'll talk about in lab next week. Here we see water as a solvent. So in this case, the water molecule will attract both the negative and the positive ions in a crystal of table salt, sodium chloride. So we see naturally all of the negative poles will surround a positively charged cation, in this case sodium, and vice versa. You have most of the positively charged ends of the water molecule will surround the the chloride ions shown on the bottom portion of the slide there. We're going to talk about doing certain things in the lab in particular where you may actually run out of enough available water molecules to actually dissolve the salt. In that case, we'll know that the solution will actually be saturated at that point. So as we said before, water is the ideal medium for any kind of chemical reaction. So we have a hydrolysis reaction. The water is added to break the bonds. In dehydration synthesis, water is removed to make the bonds, and therefore is going to be a product. So in this case, especially in the body, these a lot of times will be reversible depending on which direction we're going with the reaction. So water has very particular thermal properties. This actually looks like a very good test question. Water has a high heat capacity, which means it can withstand quite a bit of temperature before it will change into a different form. So in this case, if you were to boil water, it would take a good amount of heat to change it from a water to a gas. So it's very good for maintaining homeostasis. Now on top of that, it also has what's called a high heat of vaporization. So this will actually draw heat away, so it has a cooling effect as the sweat draws the heat away from the body, usually from exercise or fever or other kind of effects that we'll talk about once we get into the integumentary system. Also has a very cohesive property, so this can actually adhere itself to the glass. A lot of times it will dome themselves up. Also provides surface tension as well. So if you ever take like a leaf and floating on water, the hydrogen bonds actually can hold that leaf together, so it can hold light objects on the surface without breaking its tension. So water is also the major lubricant found in the body, so it's a major component of the body fluids. Also reduce friction along with some other constituents there as well. In particular, reducing friction in both membranes and organs. So in this case, like I mentioned briefly in the first chapter, the lungs and the pleural cavity have what's called surfactant. This is actually created, I believe, 32nd week when the baby's developing. So very simply, the surfactant is there to make sure when you take your first real breath in and you take the first real breath out, the surface of the lungs do not stick to each other, so therefore it expand a second time. Without the use of surfactant, basically the lungs get stuck together that first time, and of course that can be fatal to the baby. We also find water as lubricant found in the pericardial cavity, also in the peritoneum as well. So very simply, without these kinds of things, you will have different organs that would basically rub on each other and cause friction. It can be very painful at the same time. We also see this in certain muscles, at least in my practice. We see certain muscles that, for want of a better term, they dry out, or let's say they become so scarred that water cannot act very well as lubricant. It becomes very painful to move those muscles. So let's talk about different types of mixtures. So mixture is the combination of different types of elements or compounds that are not bonded together. So three types of mixtures that we see is the solution, which is very simply transparent, might have small particles found at the bottom of the container. This could be salt water, it could be sugar water, but for the most part it is still transparent. A colloid is generally opaque or possibly translucent. A good example of a colloid would be milk. Milk proteins will stay suspended in the liquid. If left out too long, then it will actually start to filter out. Last thing we see is the suspension, where the particles will settle out with a certain amount of time. So our blood is technically a suspension. So if you were to put blood into a centrifuge, you can see different sections of the blood. In this case, plasma. You would see different types of platelets, and then you see all the red blood cells and white blood cells down at the bottom of the tube. So again, solution is going to be transparent. This could include different solutions of glucose solutions. It could be a solar solution. It could be iced tea without anything else in it. Colloid, best example is milk or milk with magnesia. Suspension will be our blood, where eventually, if you let it sit long enough, the particles will settle out with time. As far as this slide goes, the only thing I really want you to take away from very simply is 1 molar solution is equal to 1 mole of solute in 1 liter of solution. So that's 6.02 times 10 to the 23rd molecules of a solute in 1 liter solution is known as 1 m or 1 molar solution. So for acids, bases, and salts, we look at how each one reacts into beakers of water. So for an acid, very simply here, we see hydrochloric acid. It will dissociate itself into hydrogen ions and chloride ions. So basically this was known as a proton donator, so it'll dissociate the hydrogen atoms. If you put in a base like potassium hydroxide, it'll dissociate into potassium ions and hydroxide ions. So this is going to be known as a proton acceptor. If you take a salt, such as potassium chloride, it simply dissociates into 2 different ions. In this case, the potassium ion and the chloride ion, so cation and anion respectively. It's this type of interaction with water that creates the pH balance. So neutral will be pH of 7, and that's going to be moles per liter. And by factors of, or powers of 10, we have an increase of hydrogen ions. It becomes more acidic. When we look at the opposite end of the spectrum in the more basic or alkaline set, basically we have an increase of the hydroxyl ions. So again, these are by powers of 10. So if we're going from pH 7 to pH of 5, it's going to be 100 times more acidic from 7 to 5, 10 times 10. If we go from neutral pH 7 up to the pH of 10, more basic, it's going to be 1,000 times more basic or 1,000 times more hydroxyl ions found in solution to make it more basic. So click through the animation here for both acids and bases. We'll explain it far better than I can. We finish with that. We'll see you in the next slide. Coming from the last animation on acids and bases found in the body, we know very simply that a lot of the fluids that we see are going to be in a very narrow range, usually going to be more so in either the neutral range or possibly on the acidic range. Very few kinds of fluids we'll find to be in the basic range found in the human body. But as we do see, each of those fluids has a narrow range where it can function properly. You become too acidic or too basic, and coming out of those two bigger ranges, certain types of reactions can no longer occur, and irreparable damage can happen as well. Fortunately, the body does have a buffer system, which is known as the bicarbonate buffer system. So basically, very simply, we have carbonic acid, which is a weak acid that the body can control. If we find ourselves with an abundance of hydrogen ions, in a strong acid, very simply, we can actually snatch up the hydrogen ions by bonding with bicarbonate ions to form more carbonic acid. Vice versa, if we see that there is not enough hydrogen ions found in a solution, in this case more basic, then very simply the carbonic acid will break and actually form more hydrogen ions and also form more bicarbonate ions, and therefore tip the scale of the pH back in neutral range. Shifting from inorganic to organic compounds, all organic compounds we're going to find in the body will always have carbon within them. Now, as I mentioned before, carbon has very important properties of forming different types of bonds, of the 4 bonds per carbon atom, to combine in a variety of different shapes. So they can form chains, it can form rings, as well as a variety of different structures. It can actually form things that can be easily stackable, such as saturated type of lipids, where you have unsaturated lipids which are very free and very fluid. Carbon compounds by themselves do not easily dissolve in water, and again, we'll test that when we get into the lab. But also they're very important because they're a very good source of energy, especially as it forms bonds with a multitude of different molecules. So let's look at some of the functional groups of carbon. Some I'll highlight a little more so than others. But very simply, the hydroxyl groups, these are your alcohols. These will dissolve very easily in water. Sulfohydryl will have a sulfide, otherwise known as a thiol group. These are very important for the structure of certain types of amino acids and help stabilize the structures of proteins, both in the tertiary and also the quaternary structure of proteins. Carbonyl groups found with ketones. These are polar and hydrophilic because it has an electronegative oxygen atom. You also have aldehydes as well. These will actually have a carbonyl group at the end of the carbon skeleton. Carboxyl groups are very important because you will find these always on one end of an amino acid chain. The other one will show in just a few seconds here, but this has a negatively charged from the pH of the body cells and hydrophilic. Very simply, this will bond with the other end of amino acid, which we assume would be positive. Here we have esters that form the backbone for dietary fats and oils, in particular triglycerides. It's also, we find aspirin as an ester of salicylic acid, very important for those who get into healthcare because it's a pain-relieving molecule. Phosphates we find on ATP, as well as phospholipids found in the cell membrane layers. These are very hydrophilic because of the negative charges found off the phosphate group. And last we have the amino group. This is the other end of the amino acid. This is the positive end, where you find a nitrogen bonded with two hydrogens. So very simply, this will link up with the carboxyl group and link up each amino acid from end to end. But again, you will always have an amino group on one end and a carboxyl group on the other end of all amino acids. So the first major molecule based on carbon we're going to find in the human body is carbohydrates. As you know, carbohydrates will have the highest amount of energy storage found throughout the human body. They follow the basic molecular formula of C6H12O6. We start with the monosaccharides. These are the simplest molecules that we find. First, we find the hexoses, which are 6 carbon rings. We find glucose, which is the main blood sugar, fructose found in fruits, and galactose, known as our milk sugar. Our pentose sugars are going to include deoxyribose and ribose, both found in DNA and RNA respectively. This is going to form the backbone of those particular chains. Now, when we take in the different 6 hexose rings, glucose, fructose, and galactose, and we combine them into disaccharides, meaning 2 monosaccharides together, we can form sucrose, which is glucose and fructose. Lactose, otherwise known as milk sugar, is glucose and galactose, and maltose equals 2 molecules of glucose together. If you were to string the disaccharides into longer strands, you have what's called polysaccharides, and these could be thousands of monosaccharides long. We find glycogen found in the human body, mostly in the liver and the skeletal muscles. It's the only polysaccharide that is produced in animals, in this case in humans. Although we can consume starch, which is found in plants and many carbohydrates in food, these we can digest. The undigestible polysaccharides are known as cellulose. Even though we cannot digest them, it does help push food through the GI tract, almost like a roto-rooter type thing. It does help kind of clean things through so nothing gets stuck or adhered to the intestinal tract. Here we see the monosaccharides. We first see the 5 carbon pentose rings. You see deoxyribose on the left and ribose on the right. We see the 3 hexoses, the glucose, fructose, and galactose molecules. And of course, as we know that the molecular formula is the same for deoxyribose and ribose, they are technically isomers, just the same as glucose, fructose, and galactose as 6 carbon rings, hexoses will also be isomers. Here we see disaccharides where both the hydroxide and hydrogen ions will combine to form H2O and the covalent bond forms. So here we see the lactose molecule formed by galactose and glucose, and maltose formed by 2 glucose molecules. Here we see a representation of a polysaccharide, each with the individual glucose monomers. So it's just not a single chain. They can also have branches all throughout. And again, these can be either tens or thousands of glucose monomers all linked together. And as I mentioned before, this could be glycogen found in either the liver or the skeletal muscle of humans. So the next macromolecule we'll examine are lipids. These are known as your fats and oils. Fats are known to be solid room temperature, whereas oils will be known to be liquid at room temperature. First, we'll look at fatty acids. These are the backbones to basically to produce triglycerides and phospholipids, or it can be broken down to generate ATP or adenosine triphosphate. We find triglycerides. These are probably the most densely packed storage unit that we have in the body. Even though glucose may be the most numerous, triglycerides by themselves hold in the most energy storage. Very important for insulation and protection. The triglycerides will be found in the subcutaneous fat layers all throughout the body. So this acts as a cushion as well as keeping our body heat regulated as well. Phospholipids, which most of you should remember from the cell membranes. Also steroids. Cholesterol being the first or the precursor to all the rest of our hormones. This is known as a 4-ring structure, which we'll look at a little later on. This is the precursor to bile salts and vitamin D as well. So cholesterol, as far as cell membranes go, if we have too much cholesterol in the cell membrane, then the cell membrane becomes less permeable, less fluid. Certain types of gases and other types of substrates and products cannot enter the cell or exit the cell properly. If we have not enough cholesterol, very simply the cell membranes can break down and basically lyse or break the cell down. Bile salts, as we know, is very important for digestion and absorption of dietary lipids. Vitamin D, very important for regulating calcium and bone growth and repair in the system of the human body. Adrenocortical hormones and sex hormones, which really won't cover too much in this course, more so for HAP2. We'll talk a little bit about the iconosoids found on the right-hand side of the chart here. In particular, prostaglandins and leukotrienes, very important for hormones, blood clotting, and also the inflammatory response, which we'll get into when we get into the integumentary system, bone, as well as some other systems towards the end of the semester. Also very important for airway diameter. So we're talking about the parasympathetic nervous system, very important for that, as well as the sympathetic nervous system. Lipid breakdown, also for smooth muscle contraction. Other lipids will include the carotenes, very important for the synthesis of vitamin A, also very good antioxidant. Vitamin E, very important for wound healing, prevent tissue scarring, which we'll talk about again as well for the integumentary system. Also works as an antioxidant. Vitamin K, very important for blood clotting proteins, and finally lipoproteins will transport lipids in the blood, will carry triglycerides and cholesterol in tissues and remove excess cholesterol from the blood. This particular structure is very important because triglycerides and other fatty acids do not transport very well on their own throughout the blood. The lipoproteins are basically a carrier to bring lipids back and forth to the cells. So here we see fatty acids in either the saturated or unsaturated state. Very simply, a saturated fatty acid has all the carbon atoms are, all the bonds are single bonds filled with the maximum amount of hydrogens. You see under the palmitic acid found on both the left and right side here, very simply, all the hydrogen atoms are paired up with the carbon atoms, so all the carbon atoms are full with single bonds. The oleic acid that you see on the left here, you notice there's a little kink there. That kink there is due to the double bond between two carbon atoms right through there. So very simply, that allows that particular oleic acid to be more fluid or to be a liquid at room temperature, whereas the palmitic acid will be actually a solid at room temperature. Because very simply, when you have the long carbon chains in the palmitic acid, these stack very easily and they adhere to each other through the glycerol molecule. So they will stack and become very solid. The oleic acid, very simply having the kink there, allows the oleic acid to be more fluid. This is very important when we talk about the phospholipid bilayer. So very simply, they do not stack very well. So they simply slide off one another and very simply make things very fluid. So here we see the structure of a triglyceride. As we know, this provides protection, almost like a cushion. Also provides insulation to keep our body heat trapped therein. And also it's a very, very densely compact molecule of energy. Very simply, we see a glycerol backbone here tied together to each of the acids here by ester linkages. We see the palmitic acid just above is saturated. We see the stearic acid also saturated, and the last one, the oleic acid, is monounsaturated because there's one bond there, a double bond between two carbons. So there could be an addition of one more hydrogen atom to saturate that particular fat molecule. If we see multiple areas of double bonds found along the acid, that is known as polyunsaturated. Phospholipids, as we mentioned before, is very important for the cell membranes. We see two fatty acid chains, very different from the triglycerides, excuse me. They have three fatty acid chains, and also the phospholipids will have the phosphate group, which we know is very hydrophilic, and the nonpolar tails of the fatty acid chains are going to be hydrophobic. So naturally, the polar heads will face both the extracellular fluid and the intracellular fluid, respectively, whereas the nonpolar tails will face inward because they're hydrophilic and don't like the fluid whatsoever. Now, what's nice about these polar heads, you can try and think of these, again, we get into the cell membrane chapter in chapter 3. If you think of a bunch of balloons in the wind, very simply, the polar heads will move back and forth all along the cell membrane. Occasionally, they may flip from the extracellular fluid area to the intracellular fluid area and face the opposite direction. But at all times, the polar heads will be facing the fluid, non-polar tails will be facing inward towards each other. Here we see the steroids. First we see cholesterol, which I mentioned before is the 4-ring structure. So very simply, we see 3 hexoses and 1 pentose. And then depending on what kind of steroid or any kind of hormone that we may create from there, they have different hydrocarbon tails. In this case, we see estradiol, which is a female sex hormone. We see testosterone on the bottom left-hand corner. And of course, we see cortisol, the major stress hormone that we see at the lower right-hand corner. But all 4 of them have the same 4-ring structure. Next macromolecule we'll talk about are proteins. We find proteins all throughout the body with doing a variety of different types of functions. This actually makes a very good test question as well. So very simply, proteins will give either a structure to the body, such as collagen, which gives us a nice little framework for bone and for cartilage. It can regulate certain processes, in this case insulin or substance P, which is a pain-producing type of substance. It can provide protection, which is very important nowadays, especially with antibodies. We also see proteins will act in muscle contraction with actin and myosin films found in all types of muscle, whether it be smooth, cardiac, or skeletal. They can transport substances, in this case hemoglobin, where the oxygen will bind with the iron. You'll also see specialized myoglobin cells, or myoglobin proteins found in muscle cells, also for oxygen transfer. And finally, they'll serve as enzymes, in this case ATPase, which basically will deal with either ATP production or ATP catalyzation. Amino acids are basically the basic components to form different types of proteins. There are 20 known amino acids, 9 of which are essential, 11 are non-essential. And what that simply means is the non-essential are what the body can produce on its own. The 9 essential are what we need to get from our diet in order to produce certain types of proteins. If we do not have certain types of essential amino acids, then certain types of processes can simply break down and also cause some irreparable damage. That's why it's very important to diversify what kind of protein intake that you're taking as well. And again, on every amino acid, you will always have an amino group on one end, which is the positive end, and the carboxyl or the negative end on the opposing side. Here's a list of the essential versus the non-essential. I just think it's important for you to know which ones are which. I probably will not test you very heavily on this. I may simply ask you whether it's simply an amino acid or not. It's just I leave this list up for you for now for your own information, but I will not ask you very simply with a list if I had histidine, isoleucine, lysine, and arginine, which one's essential, which one's not essential. I wouldn't really ask you that. I just be able to have you identify what is amino acid and what may not be. So I may ask you, say, tryptophan next to actin, myosin, and say ATPase, which one is amino acid? You know, obviously tryptophan is amino acid. Polypeptide chains, which can be either produced through dehydration synthesis or can be broken down as hydrolysis. So this reaction can go both ways. It's also reversible. It can both be a catabolic versus an anabolic type of production. Very simply, with dehydration synthesis, we can see obviously you form the peptide bond between the carboxyl group and the hydroxyl group, and you also produce water as your other product, or you can use water as a hydrolyzing action to actually break into each individual amino acid. We see this naturally. In particular, we see the breakdown of certain types of enzymes that are considered to be recycled, or we also see this with transfer RNA as we build certain types of amino acid chains to form proteins just outside the nucleus in the cytosol of the cell. And as these amino acids are paired up or form peptide bonds, we see 4 different types or 4 layers of structure to organization of a protein. First, we see the primary structure, which is simply the polypeptide chain. As these start to form, they can form into either alpha helices or turn into beta-plated sheets. This is known as the secondary structure. This is where things will start to coil on themselves and start to form hydrogen bonds to adjacent molecules, which will start to give the protein its shape. Finally, the beta plate sheets and the alpha helices will bond together and form the tertiary structure, or a three-dimensional chain of the three-dimensional shape of the polypeptide chain. And then we finally see the quaternary structure. We see two or possibly more of the polypeptide chains of the tertiary form actually form together. So enzymes in particular will have this quaternary structure where we have two different polypeptide chains will fold in on itself and actually will form itself together. So as we're talking about protein configurations, I came across this a couple years ago, which I found really to be interesting here. Very simply, scientists are having a tough time trying to figure out the actual structures of certain types of proteins. So what they would do is they would configure computer programs to try and figure out how different things are put together and how, using different rules, computers were having a really tough time trying to figure out certain structures of certain types of proteins, or how to circumvent certain types of things as far as for disease process, so on and so forth. Anyway, long story short, very simply, what these scientists had done instead was, knowing that gamers in particular, I'm a gamer myself, knowing gamers are very completist. Very simply, they'll play a game and they'll play it through once, they may play it through again, and try and find every little nook and cranny found in a game. Well, anyway, they use gamers' innate sense of curiosity and just trying different things to try and help them solve different types of anatomical riddles. So very simply, what they found was what gamers could do in 2 months. Very simply, computers could take almost 10 years to try and figure out different types of structures with folding proteins and so on and so forth. So in lieu of doing any kind of question and answer type thing for this chapter, what I encourage you to do is go under one of these three websites, very simply start an account, which is free, and very simply do a screenshot of you at least going through the first couple steps of this particular game using either Foldit, which very simply will help folding proteins into a stable structure. They already have rules already built in to the game itself, and very simply, this helps almost like a, not quite a crowdfunding, but it's like an open source type thing where you can pick up where, say, a last player left off, trying to figure out exactly how these proteins will fit into shape. Recently, over the last 2 years, I believe, scientists under Foldit have actually been able to make synthetic proteins using this particular technique. So I just thought it was a really nice way of bringing in science and also gaming at the same time, because we're finding gamers actually are a very good resource for trying to figure out certain types of puzzles, which we as humans, we love to categorize, we love to try and figure out puzzles. For some reason, it really kind of generates a lot of good feedback in the brain, whether it be through serotonin or dopamine, what have you. But so try either Foldit, or you try Tortora, which looks at RNA configurations, or you can try iWire if you like. It basically arranges nerves into a three-dimensional pattern. So what I ask very simply is try one of these three websites, or you can do all three if you want. You'll get not only credit for doing the question for this particular chapter, I'll also give you 2 extra credit points if you give me a screenshot with your account name on there. Just going through the first couple steps. You don't have to do it for very long, but just do it for the first couple minutes or so. So at least you visit the actual website. And I'll try and reinforce this again when we get into lab, both first and second week. Hopefully you guys will be watching this before we actually get into class in particular. But like I said, if you want to get some extra credit for the next exam, I'll give you guys 2 points to go towards the exam. So I'll take care of at least 1, if not 2 questions on the exam. But also you'll get credit for finishing the assignment for Chapter 2. So let's talk about denaturation. So this is where we see irreparable harm happen with proteins. So basically we see excess change in temperature or pH or some kind of chemical exposure where it just changes the protein shape. So in essence, what we'll do in lab is we'll basically take milk and add vinegar to it, and we'll basically see what happens. And basically what will happen is changing the pH will make it more acidic, will curdle the milk. So basically what you're doing is you're taking the casein and you're actually causing it to curdle or reform its shape. Another example of this would be egg whites, albumin. It's clear when you first crack into the pan, and once it heats up, it turns white. So very oftentimes there's no way to kind of get that to go back. For the most part, denaturation very simply means that it is irreversible. But occasionally with certain types of processes, some things can be reversed if caught early enough. Enzymes, as we know, are the catalyst found in the cell. Enzymes are very often highly specific, so they only take care of one, maybe possibly two types of reactions. And as we mentioned before about activation energy, it makes the actual reaction highly efficient by reducing the activation energy. It's subject to cellular control, so very simply, if we need, let's say, to generate ATP, there's going to be certain cellular functions that will turn these kinds of things on and off. Free energy levels often do not change, even with the enzymes present. Under new research, it used to be, at least when I was taking classes about 27 years ago, very simply, enzymes and substrates, it was like lock and key fit. Now we're looking to see, it's actually more, we call it an induced fit. So in other words, what happens is the enzyme, when it comes in contact with the substrate, the enzyme actually will change configuration a bit and hold the substrates in place, do its function and produce the products, and actually revert back to its original shape. A lot of times you may actually see an apoenzyme or a cofactor to help move these particular enzymes and reactions along. So here we see on the first picture here, we see the active site of the enzyme. We see substrates bind at the active site. At the second position here, you basically simply see the enzyme will change its shape. It will produce its products, and then very soon will revert back to its original shape, getting ready to accept the next set of substrates. Now this can happen 1,000 times a second, depending on what type of reaction that we're dealing with. So if you're trying to simply break down sucrose into glucose or fructose, this can happen quite rapidly. So here's a good animation for the enzyme functions in ATP. This one's a little bit shorter than some of the other ones. When you're finished with that particular animation, and come back to the next slide. Now, DNA and RNA we'll talk about a little more so when we get into the next chapter for cellular organization. But again, this shouldn't be very new information for you. Very simply, DNA is a genetic code found in nuclei body cells and regulates, I should say, all of cells' activities. These are going to be a double strand, a double helix orientation, paired up with different types of bases, adenine, guanine, thymine, and cytosine. Whereas RNA is going to be a single strand. Thymine will be replaced with uracil instead. This will help guide protein formation using different types of RNA. It's also very important for transcription of certain portions of the DNA code to form the amino acid chains in order to form certain types of proteins and enzymes. So looking at the differences between DNA and RNA, as I mentioned before, the nitrogenous bases for DNA include adenine, cytosine, guanine, and thymine. Main difference is RNA will not have thymine, but will have uracil instead. The sugars found for DNA and RNA respectively will be deoxyribose for DNA and of course ribose for RNA. You have double strands or double helix found in DNA in a twisted ladder, and RNA is a simple single strand. Nitrogenous base pairs, or the hydrogen bonds, and this is what helps keep the shape for DNA. You'll basically have 2 hydrogen bonds found between adenine and thymine, and 3 hydrogen bonds found between guanine and cytosine. You'll find the same thing with RNA, except uracil will take its place instead of thymine. So adenine and uracil will have 2 hydrogen bonds, whereas guanine and cytosine will have 3. DNA will be self-replicating. RNA is based off a DNA blueprint. As we know, the function of DNA is encode information for making proteins. RNA is going to carry the genetic code and basically make the actual proteins into amino acid chains that will fold in on itself. So we find 2 types of DNA, one nuclear and also mitochondrial. For RNA, we're going to have different types such as messenger RNA, transfer RNA, and of course ribosomal RNA. And of course, we'll finish this chapter with the last macromolecule for energy storage. Is ATP, or adenosine triphosphate. As we know, this is the principal energy storing molecule found in the human body. And as you can see, it's an adenine that's going to be connected to a ribose with potentially three phosphate groups. Adenosine diphosphate will be your ADP. When we break that bond between the last phosphate group and the next, we convert the ATP into ADP, and of course this will be an exergonic reaction where energy is released to drive another reaction found in the human body. Now just a reminder, instead of doing the question as we did in chapter 1 where we compared human and viruses, whether they're alive or not, I want you to go into one of those websites we had talked about before, either fold it, Eternate, or iWire, and create an account. Go for the first couple steps, try it out a bit, take a screenshot, and you'll get yourself 2 extra credit points, as well as credit for actually doing the assignment for Chapter 2. All right, so we'll see you for Chapter 3 for cellular organization in the next video.